Hi, I'm Marisa. I'm a scientist here at the University of Sussex. Thank you so much for taking part in the SPaRCS project. You are now the extended family of the Baxter Lab. We will be looking at cell division and chromosome stability, and your work is going to help us understand the process and how things work much better. Welcome to the family. Today I'm going to show you the methods and techniques you're going to need to carry out your SPaRCS project. This video is to work alongside your lab book, which we will provide you. However, if you do not find this video or the guide answer any of your questions, please email us. You can find our contact details at the back of your lab book. Before any lab work, it's very important to make sure you're wearing the right safety equipment. This is to protect you from any spills from your experiment, but also very importantly from any contamination caused from you. So, make sure you have your gloves on, your lab coat, and if you have long hair like me, make sure it's nice and tied up. When you finish your lab work, make sure you dispose your gloves in the appropriate bin bag, so not your normal bin. And also make sure you wash your hand very thoroughly. Now we're ready to start our experiment. It is good practice to make sure you have all the material and equipment you need for your experiment ready at the beginning, and make sure you clear off anything you don't need so you have a clear bench or desk surface to work on. Also, you need to make sure you disinfect your table or bench either with 70% ethanol or a disinfectant. You will need Eppendorf rack, at least 3 Eppendorfs, pipette tips, you might need 2 depending on which pipettes you'll be using, and your 2 pipettes, your square agar plates with YPD agar, inoculating loop, glass bead, waste beaker, and another beaker where you're going to be recycling your beads, the plates containing the yeast cells you'll be working on, hemocytometer and the cover slip, microscope, also, lastly, distilled water. Now I'm going to show you how to use a pipette. So the pipettes are used for transferring small amounts of liquids. Here's an example of a pipette. This particular pipette is from 100 microliters to 1,000 microliters. There's many different types. So there's one from 2 to 20 microliters and many other ones. It all depends which and what volume of liquid you need, and you pick the right pipette. There are many different brands of pipettes, so they all might look slightly different to each other. So please speak to your lab team to see how to use your particular pipette. But in general, they are all pretty much the same. So, to get 1,000 microliters on this pipette, you will need to twist this bit here and get to 1,000, like so. To use the pipette, first you need to get a tip. Now there's many different types of tips depending on which size of pipette you're using. The tips are disposable. That's why we have different ones, and every time you pick up a new one, and this to keep it sterile. So, to get the liquid, we go to the first clip here, and if you continue to press it, it will bring you to the second. I'm going to demonstrate using this blue liquid here. So, the first thing you need to do is to click to the first stop, place your pipette tip inside the liquid, and slowly draw up the liquid. And now you should have 1,000 microliters of liquid here, which is equal to 1 ml of liquid. Grab your Eppendorf, place the tip inside, near the bottom but not all the way in, Now press down to release the liquid, and this time you will press it all the way down to the second clip. Keep it pressed, and now you can release once the tip is not inside the epidural. Then you will get your waste bucket. To release the tip, you must press this button, and then your tip is disposed. You need to make sure you always have a tip attached to your pipette in order to use it, so never use the pipette like this without the tip. When you are happy you have all the material and equipment you need, you can now start your experiment. So to start off with every experiment, the first thing we always do is to label all your tubes. So you will need 3 epidural tubes and however many plates you will be plating. Here I'm just doing 1 as an example. So today I'll be using well type. So I will label the first epidural as well type 1, so epidural 1. The second tube is well type dilution 1, epidural number 2. The third one is well type dilution 2, and it's epidural 3. Next we'll label the plate. Same thing, but here we'll label the strain, which is well type, with my initial or your initial, and the date. And lastly, the temperature that you will be incubating your plates in. Same thing on the other side. And then always make sure you turn it upside down so that water doesn't get onto your plate, and we'll leave it on the side, allow it to dry up a little bit. Next, we will start. You should receive one of those plates from the university, and we will pick a little bit of those cells and place it into an epidural that will contain 1 ml of distilled water. So first thing first, got your distilled water. Make sure your pipette is in the right volume and you've got the 100 to 1000 microliter pipette if you have one, and 1000 microliter equals to 1 ml of liquid. So next we will get a pipette tip, gently press down a little bit. Make sure the lid is nice and tight and closed. Next, open the lid. You can either hold on to the lid or place it down on your bench like so. Now, press the pipette down to the first stop, then place into liquid. Make sure not the whole pipette is in so you don't contaminate it, and slowly draw up the liquid. Place the lid back on. You don't have to tighten it, but tighten a little bit. Grab your first epidural. Open the lid. Put the pipette on the side, and then press down. And this time you're going to press it all the way down. And keep it pressed, and then slowly take out your pipette. Now you can release and close your lid. And then dispose of the tip, like so. Next, you should also have received one of those inoculating hoops. Now you should see a hoop bit here and a stick bit here. Make sure you do not open from the hoop bit, always open from the stick bit so that you don't contaminate the hoop bit. Grab yourself one. Open your plate. Place the hoop bit slightly parallel to the plate, and you probably just need about that much cells on them. Next, get your first epindural, suspend the cells, and then mix. Make sure most of the cells are coming off of the hoop. And then place that into the waste bin. Once the cells are in suspension, you need to calculate how many cells you have in epidural 1, which contains your cells in 1 ml of liquid. This involves counting the cells using a hemocytometer and microscope. Next, you should all have received one of those hemocytometer plates. Take one out, nice and gently. You should also have one of the cover slip. So, there's two options of putting the cover slip on top of the hemocytometer plate. One is to blow on it. Second method is to get a pipette and then take out a little bit of the distilled water. Here you will see there's two rich here, that's much higher than the rest of the plates. In the center, just place a little drop on each side of water. Tip. And then make sure your cover slip is, the long end is on sort of horizontal way, and slowly place it down like so, and then move it around slightly so that it's nice and it sticks on, it doesn't fall off. Next, you're going to need the other pipette that's from 2 to 20 or 1 to 10, because we're going to need to use 10 microliter. Like so. Now we'll grab a pipette. Again, the same thing, press down slightly, and we'll get our first epidural. Because we let the cells sit a little bit, we have to mix it again before we take out any liquid. Okay. Again, press down the pipette first to the first stop. Place your tip down, slowly draw up your cell suspend. Now, be careful with this bit. You need to make sure you place the liquid between your cover slip and your hemocytometer plate. So on one side, just slowly release the liquid and press it all the way down, like so. Now that you've prepared your hemocytometer, you're ready to look under the microscope. This is the microscope we have here at the university. You might have a different microscope in your school, but the general usage is the same. This is the stage of the microscope. These are the objectives, and those are the lenses where you're looking. First thing to do is to switch on your microscope, and the light should come on. Make sure the stage is as low as possible. You can adjust the stage here, like so. Gradually move the stage up close to the controls until you start to see the grid of the hemocytometer. As the grid is only on a small piece of the glass, you may need to move the hemocytometer or stage, depending on your microscope, to line it up with the light. Once you see the grid, you can change to the 40 times objective and line up the squares for counting. You should now see round circles. These are the cells. Detailed instruction on how to count the cells are in your workbook. Now make sure you have one of those clickers and you're ready to count. And make sure you note down how many cells you've counted. Once you noted down the first grid, you can move on to the next grid. After counting the cells, the concentration of cells in Eppendorf 1 can be calculated. Now that you've done your calculations for the dilutions, we can now start doing the dilutions. The first dilutions will be taken from 10 microliters of the first cell culture you made in Eppendorf 1 and place it into Eppendorf 2. And then we will add also 990 microliters of the distilled water to make up to the final volume of 1 ml into Eppendorf 2. It's always good to start off with the larger volume and then add the smaller volume, because then you find it much easier to mix. So let's get started. You're going to need again your 100 to 1000 microliter pipette. Set it to 990. Take a tip. Again, we'll press down to the first stop and then slowly release. Place that into Eppendorf 2, like so. Close the lid, release the tip. Next, you're going to need the other pipette. It's the 2 to 20 and set it to 10 microliter. Get the correct pipette tip. Now, before you draw the liquid, make sure you mix the cell culture, as they've been sitting down there for a bit, so the cells normally will sink down to the bottom, so a good mix, it's always good. So again, press it down to the first stop, press the tip, and then slowly release. Close the lid, grab your second Eppendorf, place the tip inside the liquid, press it all the way down, keep it down, and then release. And then you can mix like so. And that's our first dilution. Next, you're ready to make your second dilution. So firstly, you will add however many microliters of distilled water you calculated into Eppendorf 3, and then add however many microliters of cell culture you calculated from Eppendorf 2, which is your first dilution. So again, get the correct pipette. Take out the water. Place in Eppendorf 3. Close the lid. the second pipette, again, depending on what volume you need. Again, mix the first dilution tube again, like this. Always make sure you close the tube and place it into Eppendorf 3. Again, make sure the tip is inside the liquid and then release. If you really want to make sure you mix it properly, just release your pipette again and then just press it up and down, keeping the tip inside the liquid. This way you can ensure all the cell culture inside the tips are mixed and released into the Eppendorf. And then again, mix it a few times. And now you should have in Eppendorf 3, which is your second dilution, 3,000 cells inside Eppendorf 3 in 1 ml of cell suspension. So now that you've done all of your dilutions, you're ready to plate 100 microliters of your cell suspend from Eppendorf 3 onto your agar YPD plate. And this time you can turn it around where the lid is at the top and the agar plate's at the bottom. So grab the right pipette. So I'm using the 100 to 1000 microliter pipette, and then set it to 100 microliter, like so. Now get your Eppendorf 3 and make sure you mix it before you draw up the liquid. Next, you can either hold on to the lid or place it down on the bench like so. Release the liquid roughly in the center and make sure your tip does not touch the agar and press it all the way down. Close the lid and then release the tip. Next, you're going to add these nice beads, and what it does is to help spread the liquid. You need roughly around 10 beads. So be very careful. Don't worry if some falls out, but try not to. Now, what you do is just mix it back and forth in one direction. Turn your plate 90 degrees again. Mix it about 10 times and then turn. And just keep mixing until you see that the plate is completely dry and there's no liquid left. Once it's all dried, grab another tube, or you might get provided a beaker, and place the beads back into this. So we always like to recycle those beads so we don't waste them. Be very, very careful. Make sure you get rid of all the beads and close the lid. And then make sure the plates are kept upside down, and then now you're ready to place it into the incubator that's set to the temperature of your experiment. So now you're ready to put it in the right incubator. Make sure it's upside down, like so. The plates are left in the incubator for 3 to 4 days while the colonies grow. So after 3 or 4 days of incubation, you can now take your plates out. After the incubation period, the cells are removed from an incubator and put into a fridge for around a week. This helps the colonies to develop their colour, after which you can count the colonies. The plates can be counted any time after the colour has developed and will keep in the fridge for a few weeks, so you can count the plate at a time that suits your school schedule. Once the colour has developed, you can count the colonies. We want to measure the number of red colonies, white colonies, half sectors, and any you aren't sure about. There are tables in your lab book that you can note down the numbers of colonies you counted. When you remove the plate from the fridge, you will probably find there's some condensation on the lid. This is why it's always best to store your plates upside down so the water doesn't get onto your agar plate and ruin the colonies. You can remove the condensation by wiping the lid with a tissue, like so. It is easier to count the plate in quarters. You can make a grid on paper and place the plate on top. Make sure you center it. Then count each type of colonies in each quarter and add these up for the plate. Make sure you note the numbers on your table. In this example, we have used the wild-type strain, so most of the cells are red. You will use a range of different strains that we will provide you. We don't actually know what the end results are. It's for you to tell us, and that's what makes this project really exciting. So let's get counting. What I find easiest is to dot on all the colonies that I've counted. So if we start with this quarter here, we can see that most of them are all red. This one has a little bit of white, so it might be an unsure, or you can count it. So you dot on the colonies and press as you count. So that gives us 10 red colonies, and make sure you note that down. And then just continue with all the other quarters. And that's it. You just completed one experiment after all of that. You will repeat this with either the same strain or different strains. And always remember to clean up your workspace after every experiment so that you're ready for the next. Thanks again for taking part in the SPaRCS project. Please do get in touch if you need any help and advice. You can find a contact in the back of your lab book. Can't wait to hear about your data. Bye.